Knock, knock. Is it on? Yeah. No, it's on. Is it on? Okay, the halftime show comes to an end. Counting down. Halftime show comes to an end. Okay, you can continue to eat. That's not the problem, but we have to continue with the program. And it's not even fully halftime because we have, I think, 9 more presentations to come before the sun goes down. So let's try to do it now. And let me just a few words before we go into the next session. Oh yeah, I don't know why I put it down here, but as you immediately figure, there's an accent here, but we had accent before, right? So like Chinese accent, a British accent, we had Spanish accent, a lot of questions from a Spanish accent, and of course, many others. So this is also a little bit a reflection of what you find in iXCells. So very international team, and we're looking forward to further expand that too. I think I heard from the speaker some Polish accent, a French accent somewhere, I think. So it's very international, and that's what we want to be, right? Because rare diseases are not just in this country. What I also like, I have to see a few observations I made when I see the numbers, right? I think Kitch had on his slide, 360 plus million people that are affected by rare diseases, right? If you just imagine 260 million people, that is kind of the US, right? People that live in the US almost. It's a little less than the people living in Europe, where I'm from. It's just a huge number. And still, I agree with Noah. The word rare disease, rare is kind of, so how I understood what he said is this is why you called it uncommon, right? So let's continue to do what we do until these rare or better uncommon diseases show commonalities, so that more people show attention to what we're trying to accomplish here altogether, right? So just to introduce myself, I'm Helge Bastian. I'm the CEO of iXCells since a year. And also everybody knows Nian Wei, so I don't have to introduce Nian Wei. But I also want to, I think Steve didn't identify who he is. Did you? Huh? What's that? So Steve is also an iXCell, of course, and he is our Chief Commercial Officer and helping to do the external communication and in improving this for the company, also with us for some time. And we have some people have an identifier, which is the little pin we have on our reverse. So if you run into those people, you can be sure they're with us, and very precious little stickers, because we couldn't find more than the ones we have that you see today. Right, so that was a long introduction to, the next session. So the third session, as I said, it's not just halftime, but almost. And the next session brings us from what we heard earlier, the real life stories, right? Which is, I think we did it last year too, starting with the real life stories. So that people really get grounded a little bit, right? In the journeys of the families, and then rare disease foundations, I think is always a good start to really ground us and say, well, this is why we're here, right? This is why even this company exists. Modeling rare disease with patient-derived cells is what we heard in the last session. And now we're moving on to talking about building bridges to collaborations with industry and otherwise to make really progress in this area. So as the first speaker in this area, I think, do I have to push this forward? We're welcoming Kent Rogers. And let me just introduce Kent a little bit. So Kent is currently the CEO of Everyone Medicines. And, but this is, I think he will explain to us what it really is. So I just don't want to get even on some eyes here to misinterpret this. But at the same time, you're also partnered at Arch Venture Partners, right? But beside that, there is a long career already, 30 years in the healthcare industry. And I just call out a few: EQ Therapeutics, Accorda Therapeutics, Merck Sharing, right? So a number of really, heavyweights in the industry. But also, as I realized, you have been on the other side when you worked for one of, I guess, what people say are the payer side of the industry. So at your time at Optum, right? So really, I mean, a huge experience. And we are looking forward to hear more about this with regard to scalable, sustainable solutions for individualized ASO therapies. So we heard about ASO earlier. As you know, iXCells provides ASO services too. So let's listen to your wisdom. Very happy to have you here, and thank you very much. Do you need this?
So thank you for having me. As mentioned, my name is Kent Rogers. I'm the CEO of a company called Everyone Medicines. And if you haven't heard of Everyone Medicines, I will provide just a little bit of feedback and background on our start. You will see the founder of our company, actually the two founders of our company, and their picture are on the wall right here, just around the corner. So there is an established relationship with iXCells already. I know I don't need to tell this audience the statistics that you see up here, but I present this deck quite a bit. This is actually part of my pitch deck right now. We're in a Series A raise, which I'll explain that as well. But what's important to understand about what we're doing is that we're committed to the smallest space within already a very small space within rare disease. And I think you're right. Uncommon is a better word. I think we all know rare disease is anything but rare when you consider the totality of numbers. It's just the occurrence is so infrequent. So we are focused in a space that was really inspired by the fact, and this is something that I've now seen coming into this part of our ecosystem with healthcare. I've been in this industry 30 years, as it was mentioned, and I have never seen anything that has been so unfortunately organized in its disorganization, if that makes sense. Parents that are raising money for their children all over the country, and this is being repeated over and over again, companies starting foundations, and this is an audience that knows this all too well, and that's not a sustainable situation. As mentioned, our company was inspired by the journey that Mila took with her mom, Julia Vitarello. She was the first patient that ever had an ASO designed for her and her unique mutation. They did this within about a year. Now, that doesn't take into account the years-long odyssey that she had to go through to find out what her daughter had. And then when she did, that there is no company researching and or developing these products. And so Tim Yu at Boston Children's actually determined that ASOs was going to be a great modality for her because of her kind of mutation. The treatment actually did help her, but she was 6 when she was starting to become symptomatic, 7 when she was diagnosed. So you can imagine a little body like that had already ravaged her. And so she unfortunately died at the age of 10. But the inspiration that came from that experience motivated Julia to start a company called Everyone Medicines to give everyone, or at least every child, the same chance at life that her daughter Mila had. And so that's what we're going to do. We are going to design, manufacture, and deliver individualized medicines. Our current modality is ASOs. We will expand into other modalities in the future. But I want to underscore this, that while there are a lot of approaches that are going about this, one of the reasons that they asked me to come in is that I don't even know how many drugs I've launched in my career, 3 dozen. And I've been on the buy side as a payer. I've been on the sell side with both large and small pharmaceutical companies. So I understand how the supply chain works. And it's not a fun supply chain, and it's an expensive supply chain. And it's one that doesn't always make a lot of sense, as you guys all know. But I do know how to navigate it. And so I intend to navigate it with these kinds of medications. The good thing, too, is that when I came in, of the companies that are in this space, I'm already got wind at my back. I've got a very experienced team in the rare space, but we also have top-notch investors. If you're familiar at all with the VC world, which I have been in the VC world for the last 2 years, Khosla, GV, and Baker Brothers are top tier. And so they're actually supporting the company and have been for the last 2 or 3 years. They are committed to the current raise, which is good. But we also have both Tim Yu and Julia involved in our company. Julia is a co-founder. She sits on our board, and Tim is a co-founder and is one of our key strategic advisors. If you're not familiar with ASOs, I'm not going to drain the science because I think a lot of folks are, but they're rather elegant in their simplicity. It's a single strand RNA that approaches basically any of the challenging mutations that are giving either proteins the wrong direction, not enough direction, or just direction in general that they shouldn't be. And so we're going to approach this with a couple of different modalities. The RNA is H dependent, which is basically our way of being able to cleave the RNA when there is an opportunity for us to actually knock down what's happening from a protein perspective that's a wrong signal, or splice switching, which is what Mila Sen, they named it after her, was. That really is where we actually design it to enable the expression of protein to be appropriate if you're lacking that expression. This as a design is not uncommon. Spinraza is a splice switching ASO, as you guys are probably aware. And then obviously in the Duchenne's area, splice switching has been a very popular approach. Now, when I first came in, we actually were, sometimes things just happen serendipitously and you just go with it, right? It's just luck. We had 2 patients that had been identified with the same Niemann-Pick C1 mutation. And they had been struggling to try to find treatment or anybody committed to Europe as an area for treating patients. It just so happened as I was coming in that they were asking if we could help them. And we had already started down the path of developing an ASO to treat that same mutation because Tim Yu had 2 kids in Boston that had the exact same mutation. So we were already creating it to see if we could do it. And as we were completing that, we made an agreement with Gosh that if you want to do this with us, then you'll be our first treatment center and you'll be our first kids. So that's when I had the fun to be able to go to my investors and say, well, hey, you've been talking about doing this for a while. We're actually going to do it. So we need to go out and raise money and we're going to build a clinical trial. I'm not going to drain this because it's actually pretty small anyway, but this just gives you a general idea that the way we're going about developing the ASO is over-indexing to try to determine safety as well as, if we can, efficacy. Is there a biomarker against cholesterol and Niemann-Pick? That's one. But the reason that we're doing this is that when you consider a regulator and the way a regulator thinks, which is not the typical way to think, and they certainly don't have empathy or sympathy, I should say, for rare disease, we need to be as thorough as possible. So we're kind of walking through the front door with the requirements, knowing that we're going to have side door arguments to why they need to be able to approve products that we will not be able to study and that will never have been in a human before we're asking for them to approve it. So how do we go about that? So the idea here is that in the US, and this does not have to do only with the Trump administration and the fire sale that is going on at the FDA and HHS right now, which arguably that's going to be distracting anyway. But the FDA is a little slower than other countries in terms of embracing rare disease. Now, while the Rare Disease Innovation Hub was formed, and that brings 2 sides of the FDA together to try to address this, the science is way ahead of the regulators. And I know I don't need to tell this room that, but the issue within the US is they're not only distracted, but most of the guidance that they're giving is for research. It's not for treatment. And while the ASO guidance they gave for a 3-month GLP tox, that is a huge savings in the drug development phase. That's for research. It's not for treatment. And so that guidance wasn't really useful beyond that. They've also given guidance for platform designation. Seems or stands to reason that an antisense oligonucleotide, it's basically 16 to 21 polymers. We're not doing a lot of difference in terms of the backbone. That is right for a platform designation, but they gave guidance that was vague. And their response to our initial conversations is, well, you've got to bring us data. Agreed. So where do we get data the quickest and the easiest? You do that in Europe. You can get a clinical trial authorization in Europe up and running. I would say breakneck pace compared to the US. It's still a hurdle, but that's why we're approaching it. And it just so happens that these 2 children that were being treated by the Great Ormond Street Hospital, that's one of the premier neurological centers in the world. Things were just kind of coming together almost too well for us to say, we have to move, and we have to move as quickly as possible. So we started to put together a master protocol, and this is a variation on a theme you might have seen in oncology, where they'll have one drug to treat multiple indications. And the idea is that they're not spending the next 5 or 10 years building out the medication. They're actually convincing the regulator that the chemistry is what you focused on. It's less about the condition, it's about the chemistry. And if we can show safety with the chemistry, if it's a severe and life-threatening illness, as you might see in so many different cancers, does it matter what the efficacy parameter is to the extent that you need to find a primary and secondary endpoint? No. It's that it's safe and it actually can show activity post-approval. So we're doing that, but we're flipping it on its head. And so instead of one drug, we'll 8, 9, 10 ASOs. We don't know how many kids that's going to treat. And I can't tell you how many times investors are like, well, how many patients are going to be in your trial? I don't know. Well, what do you mean you don't know? Well, each mutation is treating one child, but for instance, with the first one, they're treating 2 kids. Well, how many kids have this same mutation? I don't know. Well, how many in that country? I don't know. How many in the world? I don't know. We're going to find that out. But we do know that the more people do whole genome sequencing, the more we're going to find of these mutations that may be more common. The real trick is that if you have more than 5 or 10 or 15, like ataxia in Turkey, we're investigating that with Tim Yu, it's about 60 patients. I'm sorry to say that is too many. That is too many because I have to design a traditional clinical trial and go through the process to get that approved. That's not breaking the door open in the corner to show the regulators focus on the chemistry, not on the indication, and not on the primary and secondary endpoints. So that's one of the reasons why we are sticking to a very, very small population. So the other idea is that because these are such small populations, they're not all going to occur in one country. So we have started the process of working with GOSH, is the acronym for Great Ormond Street Hospital. We've been working with Paul Gisson and GOSH to find patients that have mutations that are amenable. We've already found 2 kids in Italy, same mutation, and there are several in Germany, and we've actually now visited the Middle East, and we were at Arab Health a couple of weeks ago. They are very proactive in this space, by the way, and have done genome sequencing on almost 1 million people in the Middle East. They didn't necessarily do it for medical reasons, but it doesn't matter why they did it. They have that information now, and so being able to run our mutations against their database is part of what the future part of our business model would be. If we have drugs in a library that can treat a mutation, and you find a child that has that mutation, then they need to be in touch with us as soon as possible. So the clinical trial will be across multiple countries. It'll be across multiple mutations. And so far, the regulatory feedback has actually been positive. Now, in the context of a regulator, the reason I say positive is if they don't shut you down, then you're saying I got a chance. So then if they'll say, well, what about the idea that we cannot, and frankly, probably should not have to do a GLP tox for every single ASO that we bring to you, that's a pretty crazy strategy, right? Well, that's the reason that we're going to have 8 of them. Repeatability over and over and over and over again, and talking to them about the second one, the third one, the fourth one, the fifth one. By the 5th, the 6th one, we've been in a very iterative process with these regulators. We're showing them every one of the assays we're doing, which is pretty much the same overlap with all of them. There may or may not be a biomarker, but we'll be able to explain why there is or isn't. And we will go to the greatest lengths, whether it's wearables, hopefully not burdensome surveys that parents have to complete, but any kind of baseline and post to show that there is evidence of effect. I'm not saying efficacy, I'm saying effect, because I don't know that you could ever state that the efficacy that a parent sees is the efficacy that a regulator cares about, if that makes sense. So with this, our trial design is to basically start as we will have our first 2 patients in trial in the 4th quarter of this year. We are already starting the design of 4 additional ASOs, and then we would hope to get up to 8. You know, we may have more, we may have a little bit less, but that's the target right now. And then as we progress over the next year to 2 years, we'll get CTA approval in multiple countries. This gives us a wealth of data to negotiate a corridor with regulators for a new pathway. Now, is this a guaranteed? It's not, no. When I talk to investors about what we're doing, this gives us the best shots on goal to create a brand new pathway with regulators. But by 2027, 2028, we should have enough to go for approval through EMA as well as other countries in Europe. This strategy is really basically to convince Europe so that we can then, sorry, I'm in the wrong spot, aren't I? If we convince Europe, then that means that we convince the US. But this is the vision of where we want to be. Very much like Mila, when her symptoms showed up, they were because her mom noticed, but the symptoms had been there since birth. Newborn screening isn't here today, certainly not in this country. At some point it will. And someone asked me at one point, well, what happens when you get a newborn screen that has an NPC1 mutation or a Batten's disease mutation? I mean, are we going to treat our child at that point? That's a good problem to have at this point, because we don't even know what newborn screening is going to look like in the future, or much less how it's going to be reimbursed, but other countries are doing it. So this is what would be realistic today, as the symptoms would begin, and that means the clock starts. The best case is that a neurologist would do a knee-jerk reaction. If you've got rare, random neurological symptoms in an otherwise healthy 5-year-old, run a whole genome sequence. Just run it and see what you come up with. And if you can get any kind of cellular data from the patient, swab, I don't think urine and blood were there yet, but skin samples, then you can characterize that mutation and know what modality will work. In our case, an ASO, we could sequence an ASO within hours. And if it works, or we know that it's amenable, we can design it within 6 to 9 months. That means that a child with a previously unknown mutation, and I don't say disease state, right? Because there may not be a name for the disease, but an unknown mutation, we should be able to design, develop, sequence, manufacture, and deliver within 9 months. And that just means we need a truncated regulatory review in order to get it to the patient as quickly as possible. So our business model is all inclusive: diagnostic networks, treatment centers, obviously that we're on the drug and the manufacturing side of it, and then working with regulators and payers constantly throughout the process. You guys can read this, and I'm sure it's probably something that you already know anyway, but as I'd mentioned before, the science is way ahead of the regulators, and we know that. They know it too. And I think they're starting to respond and react, but our approach, and we hope others will take the same kind of approach, walk in their shoes with them, give them a reason to support you. And instead of theory, we have to bring them clinical evidence to have the conversation. And so while this is a very costly approach, and that's what the Series A is going to pay for, we feel like that this is the best possible way to find a brand new pathway that others can follow. CRISPR-Cas9, siRNA. I mean, there's a lot of great modalities out there, but Everyone Medicines, I think, is going to be at the bleeding edge, so to speak, of making sure that new pathway exists. If not ex-US, we can certainly try to bring it back to US if they agree. That's it.
Thank you. Thank you very much. Okay, we have a little bit of change in schedule because the next speaker on the agenda is still on the road, unless I see him here. No, I don't think so. So therefore, we move on with the next. You see it here, right? Konstantina is the next speaker here, and you're here, I know, hiding. And let me just introduce you with a little bit more really good kind of background here, right? So today you are, as it says here, at N-Lorem, and you are the Senior Director of ASO Discovery and Research at the foundation. And of course, as it shows on your CV, you have been working on RNA research for some time, right? Before that, you led the RNA and technology team at Amina Therapeutics. That was also about double-stranded RNA and so forth. Before that, you also did work on rare diseases, so it's not a new area for you. But also before you went to these positions, you had a significant career in academia. And when I was, the people that prepared this for me, when I was reading through this, it's quite something here, right? So we're talking about the PhD came from the University of Oxford, but then there were some prestigious awards. One is called the Sir Henry Wellcome Reward for Postdoctoral Fellowship. And this was done in Edinburgh and in my home country in Berlin, in Germany, in Berlin at Max Delbrück Center. So quite amazing. But if this would not be enough, you also got a major award, which I think is called the Women's Science Award, which was, I think, probably sponsored by L'Oreal and UNESCO for this. So, and I'm sure there's more to find about you. So thank you very much joining us for this meeting here. Really welcome this. We heard earlier today that there were a few foundations that mentioned, and Lorem, we are known anyway, the company, right? So, but we had this in Piotr's talk, and I heard it from Michelle earlier. So there's already collaborations going on with people that are here at the meeting too. Today we hear about what you guys do when, again, about personalized ASOs in, you call it nano-rare patients, right? So we have rare, nano-rare, uncommon, and I still pledge that at some point we all call this commonality somewhere, and we're talking the same thing. Welcome, and the floor is yours.
Thank you very much. An amazing introduction. Thank you. I'm extremely happy to be here, and yeah, what a wonderful event. And for the videos, would I just mouse? Okay, that's wonderful. All right. Great. Yeah. So feel free to call me Nadina, everybody. I have an extremely long name. I'm Greek originally. So I'm leading all the research activities at the Lorem Foundation. So essentially from ASO design, discovery, and also all our core mechanistic research approaches to really reach more patients with more efficacious ASOs and in a safer manner. First of all, I would just like to take a few moments to introduce Lorem and just tell you all what we're doing and what is really our mission. Lorem's mission is to really apply the efficiency, versatility, and also specificity of the antisense technology to charitably provide ASO medicines to treat patients with nano-rare diseases, and that means less than 30 patients worldwide for free for life. And essentially the reason that we can do that, and I'm very glad that I'm hearing ASOs here since this morning, is that really we leverage knowledge and expertise that has been around for about 35 years. And you know, our founder and CEO is Stan Crook, who is the former CEO of Ionis. So we have really leveraged all this expertise from Stan Crook, his colleagues, and everybody else on the antisense space. That has been amazing. And at the same time, while being in the shoulders of these giants, we really want to build continuous research innovation. This is where breakthrough will happen, right? And our ultimate goal is one and only one. We really want to find the best ASO possible for each patient. And this is what my team and I are really working on. First, I would just like to take you through what is our Lorem pathway to treatment. So the first part is essentially, I believe this is a pointer, maybe. The first part is the research, and then we go into preclinical development, the regulatory stages, and also the clinical development. So essentially my team is initially involved in also what we call the feasibility review, where we have the application evaluation and feasibility. So the physician sends us an application on behalf of the patient, and what we essentially evaluate, and this is, we also do it initially internally, but also we have a committee of really experts that really provide either ethics or clinical development, antisense technology, RNA, all of that, to really evaluate whether the ASO technology is the optimal therapeutic strategy for this patient. And after we have this committee and have these discussions, then we really take the decision if that application will be accepted or declined. If the application then is accepted and really we think that NASO is a suitable therapeutic approach for this patient, then this is where really we get to work. And this is where the ASO discovery and optimization takes place. So the very first thing that we do is we take the whole genome sequencing. This is one of the most important things for us, especially for allele selective programs, which I will go on into a minute. And also we obtain the patient cells. So everything that we do, and because the FDA, as I will say in a moment, we have this unique guidance for ultra rare or nano rare indications. We don't have specific steps, but we essentially have a very extensive in vitro discovery step, which we really use the patient cells for. And then we go into ASO design with our in-house AI-driven algorithm. We design the ASOs, we synthesize them, and then we really start the discovery. After that, we enter for about 20 top leads in vivo tox studies. And this is essentially the rat tolerability studies where we screen for tox. And then after that, this is essentially where really the preclinical development starts, where we have GLP toxicology, and this is where the GMP manufacturing and sterile fill and finish start. After that, and this is, we hold the IND, so we draft this massive document, which is called IND, and we essentially submit it to the FDA. And as you can see, actually, I didn't mention that in every one of these steps, you have around all the numbers of how many programs we have currently in each of these stages. And the very last thing is, of course, the clinical study and treatment. And at the moment, we have more than 30 patients that we are currently treating. And we have 16 patients that are actively on treatment, and we have more than 20 INDs submitted. And the important thing is that we have this in all 4 divisions of the FDA. So just all in all, and we started really, really small. So I'm really proud to stand in here and say that we have about 330 applications submitted today. We have more than 160 patient-directed drug discovery programs. Today, 25 INDs submitted, and the 4 divisions of the FDA are supportive right now. So just to give you a little bit of numbers and where we stand at the moment and how we're treating patients right this day, this is the NLORM regulatory clinical pipeline. So as I said, the number of INDs that we have submitted and all that is more than 20. The number of patients covered by the INDs is more than 30, and the reason for that is because some INDs cover more than 1 patient. The number of patients treated is more than 20 right now, and we're going to duplicate sure because actually my team is doing a great work in discovering a lot of ASO, so we have a lot of programs. The number of ASO doses that we have administered are more than 90, and the maximum duration of treatment is more than 2 years. And the most important thing of all is, of course, that we have pristine safety and tolerability profile. And this, of course, is one of them, is the most important thing. So really the current status of the clinical programs is that we have a very pristine safety and tolerability profile. 8 out of 9 evaluable patients show evidence of clinically important benefit. And I will show you in videos in a moment what that means. Sometimes a video is more than 1,000 words, which means that we are already seeing really important improvements in multiple domains in CNS, continued benefit with continued treatment. And very importantly, this is something that it was quite surprising to us because we start very low and very modest, and then we dose escalate. We really see evidence of CNS benefit at very low doses. We also see, in terms of kidney function, because we have some kidney targets as well, target reduction and stabilization of renal function, recovery of functions lost, demonstrating really plasticity of the CNS. So for some indications that we actually thought, oh, this person might be too old to actually treat, we find out that that is really not the case. That we think that the CNS is much more plastic than what we initially thought. And in terms of ALS, because we have quite a lot of ALS patients, we really see halting of ALS disease progression. The important thing is, as I said, we have programs in all the 4 routes of administration: CNS, liver, kidney, and eye, and we're seeing important benefits in all of them. Now, what brings us to pristine safety profiles? It's really a big balance between what happens in research, what happens in discovery, and what happens in clinical development. So you really want a rigorous discovery, and this is exactly what we do. We screen thousands and thousands of ASOs to really find the one that is really the most optimal and best. So we really want to be very rigorous in that and really screen a lot in a very high throughput, high quality manner. But at the same time, on the other end, we want to be very conservative in our dosing and monitoring in terms of clinical. So what allows us to do that is a very deep and broad relevant experience in both ASO technology, drug discovery and development, clinical development and management of patients in clinical trials, and also our regulatory processes. We have an excellent relationship with the FDA. Our systems and processes are actually can be really scalable almost indefinitely. So we have really industrialized system and processes. We have multiple external committees for the preclinical data package review and clinical safety. As I was saying, this even starts from when an application comes to NLORM. It's not that we're just deciding internally. We have a lot of external people where we do a peer review. We really have a very strong commitment to caution. And also our research physicians are really outstanding. Some of them here at Radies and all over the US. We have a very close collaboration with them. And now I would just like to show you, this is one of our patients. This is a KIF1A. This is an allele selective ASO, which I will talk about in a moment. And I would just like to, this girl had a really severe disease with multiple seizures, neuropathic pain, couldn't walk, nonverbal. So I would just like to show you.
I'm ready, sis. Oh, wow. That was... Yeah, and then what?
And then when
Oh,
my God.
See?
Look at that.
get up, you know when
Oh,
you
that was
I
amazing.
like that step though, but again. Boom, yeah. Ooh.
Dad, come on, I'll teach you how
Okay.
to
You
stand up.
teach me how to stand.
Throw an alley-oop. That's an alley-oop. Easy peasy, lemon squeezy. Oh.
Yeah, it's pretty incredible. So this is just boring words to just summarize what you just saw. This girl was, yeah, she was in a very bad state. This is a KIF1A disorder. As I said, very frequent and very severe seizures. We're seeing a significant reduction in seizure frequency and severity, significant reduction in neuropathic pain. She just wouldn't sleep because she would just cry because of pain the whole day. She's regaining, as you saw, the ability to stand up and work social distances independently. She couldn't do that before. She has a significant reduction in tremors, significantly improvement in communication with family. This is something that has been communicated to us by from her caregivers, and general significant improvement in the quality of her life. She even attended a sleepaway camp for the first time in her life. And of course, we have actually just published that in 2 publications. The second one that I would just like to show you is an ion channel mutation. This is SN2A. This is a boy that has actually, the ASO that we discovered and developed was actually administered here in Radies. This is a boy that has never walked in his life and had very severe GI dysfunction as well. This is a very severe disease, extreme seizures. This is an ion channel, so you can imagine this is very, very severe. This is an allele selective ASO as well. And here's he's walking independently for the first time. Yeah, so this again summarizes what I was just saying. Reduction in seizures, he regains the ability to walk independently, decreased ataxia and choreoathetosis, and improvement in autonomic dysfunction, normalization of bowel movements, which is amazing for him and also for his caregivers as well. So all in all, I just talked to you before about what is happening with the clinical development. I would just like to show you as a whole what we do at Enlorem. And this is really our process. This is our ASO discovery and development process. It's an individualized approach and infinitely scalable. We really use, as I was saying, the efficiency of ASO technology in parallel with the FDA guidance for individualized ASOs to really provide a rapid, effective, and affordable treatments to nanorare patients. Everything starts with the whole genome sequencing and the patient cells. This is really the most important thing. This is where the ASO discovery begins, concludes that we enter the GLP toxicology. In parallel, we have to accelerate timelines, manufacturing, and sterile fill and finish. In parallel, our regulatory people at Enlorem, they start drafting the INDs and submit. We have the IND submission, IRB approval, which comes from where the physician is. And then, of course, we have the patient treatment. For the last part of the talk, what I would like to do is to take you through what my team and I are doing, and how do we end up with that optimal ASO? What does this look like? So this is essentially our process. But before that, I would just like to take a step back. And as I was saying, I'm extremely happy today that I heard ASOs multiple times. And I think the reason for that, the reason I think that this modality is really an ideal one for nanorare patients, and also importantly, why we do what we do at Enlorem and why we do it so well, is essentially because of this particular modality. For example, if you were to take, the really big guys at drug discovery, which is essentially historically, right, the small molecules and the monoclonal antibodies, you would know that the rules of engagement are not as well understood. Whereas with ASOs, with RNA targeted, we know exactly what are the rules of engagement. We know that in this particular case, the receptor, if you like, is the RNA. So you know exactly what you're going to get. After you design an ASO, you know what it will do. It will target that mRNA, it will hybridize like a zipper, and then it will degrade or it will block something. So since then the rules of engagement are very well understood and easy to use, it really means that we can also learn from previous drugs. Not everything is just like, sometimes with small molecules, we say change the methyl, change the drug. In this particular case, we have a broad transferability. The cost of drug discovery here is modest compared to other routes of drug discovery. The rate can be months. It is very, very selective. As I said, this is an RNA. This is just based on base pairing and hybridization. Very versatile. A lot of different mechanisms here that we can do. We can alter the RNA in many different ways. And also the important thing, and this is what matters, right? When you want to do breakthroughs and when you want to do things faster and better and safer, is really to advance the technology. And with ASOs, there is an extensive rapid advancement. And this is what we strive to do. Very briefly, and I don't want to bore you too much with all the science stuff, but a lot of people talk about gapmer ASOs, right? This is the RNase H1 ASOs. This is literally what is the drug. This is literally the ASO. And a gapmer ASO, this is what is used when you want to reduce or knock down an mRNA target. And essentially, we have what we call the 5-10-5 modality. So this is where you have the 5 bases at the wings at the very beginning there with the 2 black, which is RNA bases that are essentially or modified with methoxyethyl. And then you have in the middle there that orange part, which is the DNA. This is essentially where all the magic happens, because this is exactly what provides the substrate for a beautiful enzyme, very, very selective, which is called RNase H1, where when this orange binds your mRNA target, this is where the cleavage happens. And this is how you reduce the target. Now, you can do that in what we call a non-allele selective way. So you can reduce both your wild type, the good copy, but also the bad copy, the mutant, and that is what is not an allele selective. But the beauty of this enzyme also allows you to do it in an allele selective way, which basically means that you can leave your wild type untouched, but you can only target the mutant. And I will show you in a minute how we do that. Now, this is what we use when we want to degrade, when we want to reduce the target. We also have the other class of ASOs, which is called the steric blocking ASOs. This is an 18-mer, and as you can see, there is no orange here, everything is black. And the reason for that is because you essentially have only RNA bases, that they're all modified, the 2' methoxyethyl. And here the backbone is phosphorothioate. And something that I forgot to mention before is actually, you might ask, why do you need all of these weird chemistries? Why do you need to chemically modify the backbone or the RNA bases or whatnot? The reason for that is because this allows you to have a longer duration of action of the drug, less immunogenicity, and also higher binding affinity with the RNA. This is the reasons why we chemically modify ASOs. So you have different classes. So this would actually, you would sit back. So for example, when we receive an application for a patient, we sit back, we see the disease, and we're thinking, what is the best strategy for that patient? Do we want to reduce something? Do we want to reduce both the wild type and the mutant? Do we only want to reduce the mutant? Or do we want to block something? Do we want to use a different isoform? Do we want to alter splicing? And the reason that we can do that is because really we have, the ASOs are very versatile. So as I said, we can do an RNA degradation. These are the first two in here, either with RNase H1, ASOs, or you can have the siRNAs with AGO2, albeit a little bit more of a promiscuous molecule, not as specific. But also you can have mRNA modulation. So the splicing ASOs that I just showed you before in here can be used to either alter splicing or affect mRNA stability or switch between different isoforms. So this brings me to the allele selective ASO approach. And this is both of the ASOs that I showed you in videos before, they were using these kind of ASOs, and we use them a lot in a lot of diseases for the CNS, because essentially you don't want to completely reduce the protein, but you really want to keep one good copy, but get rid of the bad one. So the way you do that, and this really leverages the specificity of the RNase H1 enzyme, is you really try to guide the RNase H1 to target only the pathogenic mRNA without with leaving intact the wild type. How do you do that? We have these little signs in a gene that they're called SNPs. You might have heard of them, single nucleotide polymorphisms. And this is why the whole genome sequencing is so important to us, because we take the entire gene and we really see where these SNPs are. SNPs are basically positions where your wild type and your pathogenic differ. And this is what we need, because we need to find these little differences so that we guide the ASOs in these positions. So the ASO can specifically, the RNase H1 can specifically degrade the pathogenic with leaving the wild type untouched. Now, we've done that. So what I've shown you is we've decided on the strategy. What do we want to do? Do we want to reduce? Yes, no. Do we want to block something? So after we have done that, then we are ready to essentially design the ASOs. And in order to design, we have our own in-house ASO design algorithm, which essentially really is the one that makes our ASO discovery highly efficient. And AI is a buzzword right now. It's amazing. The thing that is important with AI is what kind of data you bring in. And the kind of data that we bring in our design software is essentially, we're taking all of these millions of ASOs that both us and also ASOs at Ionis we've been dealing with, all relevant cell and animal models, hundreds of thousands of patients, continuous learnings from the whole genome sequencing, because it's essentially what we are striving to do, to design better and better allele selective ASOs, and designing ASOs to reach more patients. And also at the same time, of course, there's so much information out there that is amazing with the technology, and there are a lot of multi-omics data sets that we're essentially putting in our algorithm to really make it better and better. And I'll just finish the talk with just showing you, how does it really look like? You know, when you screen, how does it look like? What kind of graphs are we seeing? So our process is essentially the very first part, and this is where it takes some time, is the primary ASO screen, because we screen about 500 to 1,000 ASOs. And for an allele selective approach, as you're going to see here, is the blue is the wild type allele where you want it almost untouched, and the red is the mutant allele where you want it reduced. So you can already see that there are some patches there. Oops. There are some patches there that we have some really, really good ASOs. And we have very important and very strict minimum criteria. So things that do not pass our criteria, for example, of 80 or less than, more than 80% of target reduction, it won't pass to the next step. The next step is really the epitome, as I would call it, of drug discovery for ASOs, which is essentially the dose response evaluation, where we do a 5.4-fold dilution to really scrutinize the efficacy, potency, and also allele selectivity of the ASO. And here what we take in this step is we take about a 50 to 75, so the best of the previous step, and we essentially scrutinize what is the IC50? This is what defines the potency. What is the IC50 of the mutant? What is the IC50 of the wild type? What is the reduction of the mutant? Do we have the wild type untouched? What is the selectivity that we see? And here you can see here, they are in the red box, some ASOs that really stand out in this particular case. And finally, as we move down through the discovery path, we also do an in vitro, of course, of target analysis. This is initially done in silico, but then, of course, we also do that in wet lab. And we do that as many as necessary to make sure that we eliminate any off targets. And finally, of course, we're going to do the in vivo talks later on, but you really want to check, you wouldn't put ASOs that you know are not safe into animals. So there is an assay that has been established for years now, which is called the BJAB assay, which is essentially is a very good indicator, is done in B cells, which is a good indicator of innate immune activation. So the ones that really activate immunogenicity are, of course, thrown away. So you have essentially surrogates for that in a qPCR. And finally, you can see these really fancy robots that we have in the lab. And this is what allow us to really handle about 90 patient programs at the moment, even though we don't have a very big team. So this is all liquid handlers for high throughput screening. So as I come to the end of my talk, these are the conclusions. So we have a very highly experienced and functioning team in place to discover and develop optimal ASOs. We have the capabilities to do this rapidly in a cost-effective and high throughput manner. Our clinical experience is outstanding at the moment. The ASOs given via multiple routes of administration are safe. Our clinical benefits are seen across all organs, and very importantly, at low doses and really early in treatment in CNS. And we're really treating these patients today, and we are extremely proud and happy about it. These are all the publications to date. We try to publish as much as we can to have all of this out there. And really, thank you.
Okay. What can I say? This is Constant. Where is she? There she is. So Konstantina, this is just amazing, right? I mean, what else can I say? You called it outstanding. It's amazing on all levels, I think. And to see kids walk that have never walked is so stunning, right? So we again are reminded why we're doing what we're doing, right? So you said you also like to talk about ASOS, generally speaking. You like the other people talk about ASOS too. And I think we can promise that we will probably hear more about at least RNA modalities in general. And he arrived safely. Hi, Jeff. So are you also blown away? Still to breathe, right? Because now it's a little bit of a competition here, it seems like, right? So many people know Jeff Milton, right? Jeff is not just the founder of La Jolla Labs, but also run it as a CEO. And if I saw the news correctly, now you're switching with your co-founder and focus on technology, and she does the CEO and has to deal with all the HR issues and so on. So I think it's a relief, is it? So yeah, so for those that don't know La Jolla Labs, and he will talk more about it, a company that also thinks about RNA therapeutics and modalities. So, but it's not the first time he does it, right? So you have been working with, as a head of data science with Arcturus Therapeutics. You have been also working, of course, as a director of functional genomics and drug discovery at Ionis, right? And then before that, you also had time with Genentech. And so we know you, right? We work with you. Neon Way has worked with you before, and we're looking forward. What are the news? And yes, so Jeff, actually, I just also have this on my list here. You are a graduate from Mellon College of Science at Carnegie Mellon University. Yes, okay. So welcome again. Glad you made it safely, and here we are.
Great. Thanks. Yeah, so it's going to be a continued talk on ASOs, but this is actually a talk I've wanted to give for a long time, and it's a work in progress, because what I wanted to actually talk about was actually more about the modalities that are used. One of the things that I experience a lot at La Jolla Labs. So a little bit of history. We started La Jolla Labs. My co-founder and Tamar Grossman, started La Jolla Labs about 3 years ago. We're both from Ionis Pharmaceuticals. We were at Ionis when Spinraza was made during Milasen. So there's a lot of overlap. In fact, you're going to see some slides that I have that Stan Crook gave me that were just shown in the preview. But that's fine. That's good. But in any case, there's a lot of overlap. And so I'm going to come at this from a point of view of a company. How do you run a business doing rare disease? It's very difficult. And so every time when I get into this thinking about drug discovery, I think about it from the point of view of how can we make a business out of it? That breeds innovation that hopefully kind of perpetuates and makes it sustainable. So that's kind of the angle. This particular talk is really because I've always wanted to talk about the different modalities of drug discovery and the context of rare disease, because rare disease, you have to think of it from the point of view of rare disease and not be sort of stuck with a technology trying to fit it to the disease. So this is part one, because I'm going to talk about it in the context of chronic dosing versus gene therapy. So I'm not going to talk about gene therapy, the one and done type scenario, because that would be part two. But you've seen this before, rare diseases, it's a long tail. And what that means is that from the population, from a patient perspective, there are a lot of different individual patients that you have to focus. So you have to be more individualized. And that creates a very different landscape. The conventional approach to therapeutics development and even clinical trials is totally different. You have to interact with patients. You have to talk to families and kids, and there's no Hippocratic oath when you're in business. So you have to ride this very fine line between what's right and what's wrong, because you have to answer to the patients. Ultimately, that's what you have to do. But you also have to make a business. So that's the struggle, is how do you really do that? And when you think about the fact that 90% of drugs fail in sort of the common approaches to clinical trials, 90% fail, that doesn't work for the business that we're in. So there's a lot that has to be taken into consideration. So I get parents talk to me all the time, and they say, we want to do an ASO. And because it's a rare disease, and they've heard about it, and they think about the technology, and I say, I don't know if that's what you want to do. It's probably about $700,000 from start to finish, if you're lucky. And so what I say is, try to look at repurposing. So is there a way to repurpose a drug that's already out there? So there are companies that do this. One of them is, I'm blanking on the name, but it's Unravel. If you're anybody familiar with Unravel? So they'll for like $15,000, they'll do a screen for a particular disease, and then they'll find other particular indications that might be linked to it. They have a business model that somewhat works. I say, go to Unravel first. It's cheaper and probably faster. The tox profile, the preclinical work is super, super expensive. And a lot of people who do rare disease don't even think about that part of the work. They kind of do proof of concept, they get excited, and then they get into the preclinical side of things and the money runs out. And so you end up with a lot of dead end projects. So let's talk about small molecules. So small molecules, I started this industry when rational drug design was kind of the approach. It was kind of the, Gleevec was the idea of taking a protein from the PDB and finding the pocket that you want to get it into and designing the small molecule into that little pocket and rationally design the drug to fit into the target that you want and make it do what you want it to do. But ultimately, is it really rational? There's a lot of space you're playing around with there. Maybe AI can help, but as we're finding out, there isn't enough data to really figure out, to really kind of make that happen in a super efficient way. It's still 10 years to do a drug and about $1 billion. So that's kind of the small molecule approach. Can it be selective? Well, here's one that's actually pretty selective. So this is a drug by Merck, and it interfaces or interacts with this MET kinase inhibitor, so a mesenchymal epithelial transition layer, and it's very, very selective. I think it's like 10,000-fold more selective than any other kinase. So it's very selective to that particular target. So that's still a very costly enterprise if you go down that path. So what does that look like? So for a small molecule world, this is kind of what a typical small molecule approach would look like. You'd have a big or a large library of 1 million different compounds. In this particular case, the screening library is what's called a fragment-based drug discovery. There are other types of small molecule libraries, diversity libraries, things like that you can do these millions of compounds of screens. And then you find 500 hits for the particular target you're looking at. First of all, the target has to be extremely well characterized. And then maybe you can do crystallography to figure out how that fits in there. And then you can do some rational drug design, some med chem to figure out, I've got to make it look more like a drug and have the properties of a drug so that it sits around and does the actual, what it's supposed to do without being cleared too quickly, that type of stuff. But again, you're still looking at, in this particular case, I think this was about, so this is under 10 years, so that's quick. So this is kind of giving you an idea of the benchmark that we're dealing with a small molecule. And then this one is actually, this will be the last one. So this is from Vertex. And this is the ETI drug for cystic fibrosis, or I'm sorry, CFTR. Yeah, so cystic fibrosis. And what's cool about this is actually, this one is actually, it binds to a lot of different, it's a folder enhancer and potentiator. So you can kind of look at the phenotype of this and screen for that. So that's kind of cool. And this was actually pretty quick. I think it was less than 10 years, but they have these nice cryo-EM structures over here that show the structure. So that's nice. So the one thing I want to say about this is that there are a lot of off-target effects with this. So there's liver tox, there's kids get cataracts with this. So that's the other problem with small molecules, is that you can get very promiscuous hits in a sense. So now we go to antibodies. So this is a separate modality. This is much bigger structure. You have the variable region, the antigen binding region, and then you have this very conserved region here. So this is the part that you're going to bind to the actual, whatever it is, the molecule you want to bind to. And then there are all these different modalities that you can chop up and use separately. So for example, there are some enzyme replacement therapies that use just this Fab fragment as a delivery vehicle. So they're very versatile, but very complex. And this is one of those structures that gets into sort of downstream costly effects with synthesis and characterizing structures. So in any case, this gets very complicated. I'm not going to go through all the details, but they're very versatile in the sense that they can be used as delivery vehicles. So in this particular case, you've got an antibody with a linker and then a payload. And so for cancer purposes, that's great because you can have the antibody bind to the tissue, the specific cancerous tissue, internalize, and then the payload is the toxic part for the tissue. And then you can have something like an antibody bound to an siRNA or an ASO. And so these are structures from Avidity. I think I took that from their website, but that shows how you can go even more selective. So you can be tissue selective and then target selective, RNA selective. So that's kind of gives you some of the idea that antibodies go a little bit further as far as selectivity than small molecules, but there is a very big downside to antibodies, and that is the cost of some of the synthesis and some of the downstream preclinical studies. So when you scale up these reactions, they can be very expensive. I mean, so I was at Genentech for many years, and I used to go into the scale-up facility and talk to some of the guys there, and they were saying, I'm about to run $1 million reaction, so don't talk to me. Because if they mess up, that's it. So in any case, there's a lot of complexity that goes into this. And when you're doing things like making or manufacturing this, you have to characterize what they look like. If it's an antibody drug conjugate, these things are actually in solution in ratios, and you have to kind of find ways to develop the assays so that you can actually characterize what those structures are, so you know what you're delivering. So that's an extremely expensive downstream enterprise, but it's very selective. And so, of course, you know, this is kind of a dump of all the different things that you need to do for taking a drug, antibody from beginning to end, from cells, from sort of research all the way through to the IND filing. But this is the type of stuff that gets very costly that people don't see. So part of the reason why I love RNA is because it's targeting the genetic cause of the disease, and it's very simple as far as the modality itself. So I'll talk for the rest of the talk. I'll talk about the RNA sort of landscape, and this kind of gives the history of it. So Ionis started in 1989. And so right about here, it was started with $7 million right here in Sorrento Valley by Stan Crook, the founder of Lorem. And they went through a lot of ups and downs. It was not an easy road by any means. But in any case, Stan persisted and really worked this out. And you can see that really by the number of publications. He's very he publishes quite a bit on this, and so it's you can really kind of follow the whole path that he did for this. But the first drug was actually Formotersen. It's off market now, but this was for it was for HIV patients who had a viral infection in their eye, and so that was the first drug. And I think that was a straight DNA. I don't think that was chemically modified. It might have been chemically modified, but very little. And in any case, and then we go, yeah. So anyway, I'm gonna skip through some of that rest of that stuff. I don't think they have Spinraza up there, but Spinraza was 2016, and that was the first blockbuster ASO drug. So that was, and that is a steric blocking drug. I'm not going to go through the mechanism of that, but that was it wasn't a gapmer. It was basically just a molecule well, I'm getting too deep into the science, but basically it's a steric blocking ASO. So it's fully modified ASO. And so these are the two RNA targeting drug modalities that are typically they're talked about. So this is the Ionis approach, and this would be the Alnylam approach. So the siRNA versus the ASO, single-stranded ASO. And so the mechanism of action is basically for the gapmer ASO is it binds to the target RNA. The RNase enzyme recognizes this as basically not supposed to be there, and it cleaves it. And then you have the RNA degradation. And then the siRNA is a little bit different, but it's the same basic mechanism. And they can be viewed as catalytic in the sense that these molecules actually don't get cleaved right away, but they can go and bind to other RNA targets. So, and the big difference here is that this happens in the nucleus, whereas this is in the cytoplasm. So the siRNA doesn't actually happen in the nucleus. And so there are some, that's part of the reason why, so there's some advantages to the RNase approach when it comes to cleavage, because you can cleave in the intronic space versus the siRNA has to be on the mRNA. And so the siRNA, and in this particular case, actually this is the steric blocking approach. So for the steric blocking approach, it's the same type of molecule, but it can actually get into the nucleus. So for steric blocking, for things like exon skipping, you might have seen with DMD, this all happens in the nucleus. And so you can see that it can bind to various locations in the intronic location and cause various splicing factors that would either enhance an exon or skip out the exon. And that's the mechanism of action of Spinraza. And so here's the stand slide. So there's basically a lot of different approaches to RNA, this particular modality. And this is really the reason why we really like it. So you can do the RNase approach, which is the gapmer approach, the siRNA targeting RNA, and then nonsense mediated decay, which in a second, I want to show you a little, an experiment that we did that we really, one of the reasons we like the nonsense mediated decay is because we can use a fully modified ASO. It's not a gapmer, has a longer half-life in the CNS, and it's much more durable compound. So we could potentially use it systemically or in muscle. And then there's no-go decay, which is basically pausing the ribosome so that the body says, okay, this is not working out, so we'll degrade this. And then translation inhibition and alternative polyadenylation. I'm not going to get into that, but what we have done at La Jolla Labs, and then there's microRNA strategies for increasing expression. This is kind of like this is essentially what we're going toward, is trying to build a palette of experience for being able to do any of this work. So we've done upregulation with microRNA inhibition and enhancing canonical splicing. And don't think I'll show that one, but we have not been able to reproduce this, although it has been published at Ionis. And then we have downregulation. We've done RNase. Turns out that's a much easier process than our favorite, which is inducing NMD, which I will show in a second. And then we have done a pretty good allele selective knockdown, which is the n-lorem approach. And then we have done exon skipping and cryptic exon modulation. Protein engineering, I don't think I'll show that, but what we're able to do is essentially move a splice acceptor site so that we add amino acids into the active site of an enzyme. And we think that would attenuate that enzyme and essentially knock it down. So that's the way we would do protein engineering. So this is an example of an NMD. One of the reasons we started La Jolla Labs was because of a disease called Lafora. I've talked about this before, but this is a glycogen storage disease. These are kids that are high functioning into their young adolescence and then start a cognitive decline and end up dying in a vegetative state. It's a terrible, terrible disease, very rare disease. So what we have, and this is our software, our La Jolla Lab software on the top here. This is an AI model that we have that shows us how to splice out an exon, essentially. So what you see with this dot right here is, this is an exon, this line. The bars going up are attributes, we call cis-regulatory attributes that are associated with the inclusion of that exon. The bars going down are for the exclusion of the exon. So we would put our ASOs, which are steric blocking ASOs, on these blue dots. And those are hitting the bars that are going up. So we want to cut the exon out. So we know where to place them. And so I think we had probably about 30 ASOs for this one because we knew where to put them. We cut out the exon. That caused a frame shift, and that induced nonsense-mediated decay. And so we did this in a dose-responsive way. And then we showed it with protein. And then we also did a phenotypic assay where we're actually knocking down glycogen the way we wanted to. So what we did was we created a molecule that was fully modified so that it wasn't consumed by nucleases. So we didn't want the Gapmer approach because that has a shorter half-life in the CNS. So this, theoretically, the patients would be dosed once, twice a year. And it has that durability with this particular approach. So it's the same molecule as Spinraza, just a different sequence. The same chemistry as Spinraza, different sequence. And here's an example of what that looks like. So this was really the reason we decided to go this route, was because Spinraza has such a long duration of action. So we wanted that chemistry, just a different biology. So the last part of the modality talk is really where RNA is going, which I'm kind of excited about. I mean, there are these ADA-imers, which are the RNA editing. For the first time just recently, we went into the clinic with that. Wave went into the clinic for this particular target. I think I've had probably over the course of the last year, 2 families come to me with single nucleotide mutations in acceptor sites of splicing acceptor sites, which could be done with, they could have fixed it with this type of approach. But it was so new that we're not prepared for it. We're not ready for it. But I think that's coming. So I think this is kind of an exciting thing to see. But the other thing is, does this, we're kind of waiting to see what Wave kind of reads out and how this goes. But the other thing is also, it still has the same delivery problems that every other RNA modality has. And we'll have to work through that. But in any case, they have this prism chemistry that they talk about that I guess makes it very durable. It is a larger molecule, so it has a sequence space that targets the actual RNA, but it also has a part of the molecule that recruits the enzymes that does the base switching from an A to an I or A to G. And so they actually show some pretty compelling data that shows that they're actually getting this in vivo. and then they have some pretty good clinical data coming out here pretty soon. And so I think there's a lot more that's going to be happening in the RNA space that I'm really excited about. And so this is what the RNA editing space looks like right now. So there's a lot of stuff in preclinical, but Wave now has that first clinical in phase 1 right now. And I think they did read out some positive data on that. But ultimately, when we're talking about rare disease and the approach, it comes down to a combination of how much money, how much time, efficacy, safety. Money and time are basically the same thing, but you've got to build that therapeutic window in a short amount of time with a certain amount of money. Is if you can get a situation where you have multiple shots on goal, that's the best. If when I see these families come to me and say, we want to do an ASO, I think, that's one shot on goal, and it's probably not going to work out. It's going to take a long time to get to that point. And they're putting their own money into it. I try to kind of manage that process. Because ultimately, when you think about just the manufacturing costs, anytime you go from, I like to look at this from the point of view of chemistry and biology. If your manufacturing process involves biology, it's going to be expensive, no matter what. That's just the way it is. And in chemistry, it just has a little bit more of you can characterize the compounds a little bit better, and you can manage it a little bit better. But this is the landscape of what things look like. I did my best at kind of figuring out these numbers, but for the most part, if you can find a small molecule approach, that's the best. And then ASOs are next, and then goes from there. But this is part of the reason why you see, if you look at a lot of the companies that are really in distress, you'll see a lot of these are in the cell gene therapy area, because that late stage price tag is astronomical. And with that, thank you very much.
Yes. Okay. Thank you, Jeff. I think it shows one more time how sophisticated these RNA modalities are, right? And why there's more than one company involved in this, because there's a lot to find out and to gain too, right? So we're coming to our next speaker. Let's is Yael on? Is she on yet? Are you Yael? Dr. Weiss, are you on right now?
On, and I'm having trouble sharing my slides. It says someone else is sharing.
Okay, you have we have a few, one more minute because I'm going to introduce you first, and it says on the slide anyway, coming soon, so we are good.
Okay. You
so
set me
let
up.
me.
Thank you.
Yes, so let me introduce our next speaker. So where do we start? Because there's a long career already, and maybe the latest, we start with the latest, and while Dr. Weiss is speaking to us. So Dr. Weiss founded Masi Therapeutics, a venture that is dedicated to delivering therapeutics to patients with underdiagnosed, and here's the new term, ultra rare genetic neurodevelopmental disorders. And I point this out because we had rare disease, we had uncommon diseases, we had nano rare, now we're talking about ultra rare. And I still hope in a few years we find, as I said many times now, there's commonalities between all the different diseases. Anyway, so we will also learn that later on what Masi Therapeutics, their approach, how to address all this. But before we come there, so I just want to mention also that Dr. Weiss has been having roles at Genzyme, Merck, Ultragenics. So this goes over 20 years back. So a long career here already. I also don't want to miss to say that she is a distinguished member of the NIH-driven Bespoke Gene Therapy Consortium, the ASGCT Translational Committee, N equals 1 collaborative, and holds the prestigious title of, and this is from 2022, Termere Fellow. The activity serves, she also serves as a board member for a number of organizations like ADNP and FOXG1 Foundation. The long career started with graduating from Hadassah, and I'm sorry for the pronunciation, medical school at the Hebrew University in Jerusalem, and she holds an MD and a PhD. And this will all lead us into her talk, revealing a little bit how Mazi Therapeutics goes about developing gene-based therapeutics for genetic disorders. And I understand this will be, you will talk about your joint efforts that bring biotech, academia, and patient groups together. So looking forward to your talk, and the IT people here will make it happen that your slides will run properly. We can see you now. That's good news. And hopefully you can see us, and your slides should be up.
I'm just transitioning to slideshow.
Okay.
Great. So can you see the slideshow?
Yeah, it's not on presentation mode, but
Not-
maybe there's, if you press it on presentation mode, we should be all good. So the right on the bottom. Here we,
No, just a sec.
oh, this is you.
I dared to do it. Just a second.
We have you on the screen right now, and your slides are behind you. Can we help?
It's not moving to presentation.
Oh, don't worry.
Let me try again.
We hear you well. We see you well.
Okay, tell me what you see now.
Maybe it's loading. It was there before.
I know. Here we go.
Here we go. Yep.
Great. So, hello, everyone. I apologize for not being there in person. One of the reasons that I'm not there in person is that this morning I was part of a meeting with the FDA, with the American Society of Cell and Gene Therapy, which we actually thought would not happen, which would have allowed me to come to San Diego in person, but it did happen. And even though Kent says that the regulators are slow and not in tune with what we're trying to do for uncommon or rare diseases, we actually had a really good meeting with them on the topic of gene therapies, and a lot of openness to creative trial designs, to creative approaches to endpoints. And basically what the team that was on the phone told us was that we should talk to them as much as possible. So come to the FDA when you have ideas, ask for a type D meeting or a type B meeting or whatever meeting is appropriate for the question that you're planning to ask. And they will do as much as they can to accept your ideas and at least have a discussion on them. So that was really a good way to start the day. With all the news that we're hearing about what's happening in the FDA, it was actually a very positive meeting. So Jeff set me up for a lot of the slides that I have, so I will go through them quickly, some of them quickly, not to repeat what Jeff's already said, but I do want to talk a little bit about the Mazi perspective and how we approach working on therapies for, I know I'm adopting your terminology, uncommon diseases, and I would say very uncommon diseases. And a little bit about the lessons learned from what we've been doing over the last few years since Mazi was started. So the key messages that I'd like to convey is that the more genes are being associated with genetic disorders. Obviously, we started with 7,000 a few years ago, and now we're over 10,000. The more genes there are, the more patient-specific and gene-specific organizations there are. And gene drug development in general, as Jeff said, there's a lot of failure. It's not for the faint of heart. I have a lot more white hair on my head now than I did when I started out on this journey. But we really, in the case of rare genetic diseases, I would argue that we have an advantage if we fully understand the biology, but it's not sufficient or not mandatory. And you can choose a modality that allows you to go ahead with the therapy, even if you don't fully understand the biology of the disease. For example, gene therapy, you need to know where you need to replace the protein, but you don't necessarily need to know exactly what the protein does. Proof of concept is only the beginning, and I'll talk a little bit about what we consider as proof of concept, and then why is it so important to work with advocacy groups, and why is this triangle of industry, advocacy groups, and academia so important. So the topics that I'll cover are a little bit about advances in genetic diagnostics. What is a target product profile? So when we go to investors, when we go to regulators, when we go to payers, we need to show what the drug that we're planning to develop is going to look like. What is it going to do? How many times a day is it going to be administered? What the toxicology profile of it is. There's actually a whole list of things that we need to address before we even start thinking about going into clinical trials. So I'll talk a little bit about that and then about the teamwork that's required to get there. So I talked about the number of genes. We work in the neurology and the neurogenetic space. So a lot of the examples that I'll be giving you are from this space. And for example, when you look at the number of genes that were known for epilepsy 15 years ago, there were very few genes that were strictly associated with a specific epilepsy clinical diagnosis. As you fast forward to today, there are hundreds of genes that are associated with epilepsies and very specific genes. And these could be channels, they could be secreted proteins, there could be nuclear proteins, transcription factors. There are many different types of proteins that actually, when they're mutated, lead to seizure disorders. And the genetic testing is a key for many of these disorders that are just being associated with a gene, that it's key for commercial interest. So many companies are sitting back and saying, okay, we will wait till there are 10,000 patients and then we'll start developing a therapy. Other companies like Mazi, we say, okay, we're going to start to developing a therapy, and hopefully by the time we get to the clinic and by the time we get a drug approved, there will be enough patients to make it commercially viable. So this is Mazi in a nutshell. We're a small team, we're only 15 people, but all of us have a lot of expertise and experience with rare disease drug development. And the four bubbles, the four green bubbles exemplify our key partners in the journey to develop therapies. So we start with the advocacy groups. And this is where Mazi started, when I realized that a lot of advocacy groups, and some of them you heard from this morning, are developing therapies, spending money on getting proof of concept in cells or in an animal, and then kind of getting stuck because, as Jeff said, it's really expensive and it requires a lot of expertise. So we decided to start Mazi in order to be able to take programs that achieved proof of concept in an animal or in cell lines or in organoids and take these further into the clinic, take them into tox studies and into clinical studies. We work with the academics that actually conducted the work for the advocacy groups, because most advocacy groups look for the person that's an expert in their gene or in their protein and go to that person and ask them to start developing a therapy. So this is where the actual data will come from. And then we work with diagnostics companies because we're constantly looking for more patients for our disorders. And we work with CROs and CDMOs that execute a lot of the work that we're planning in order to get into the clinic. I'm giving SMA as an example. Jeff gave it too, but I'm trying to give it from a little bit of a different perspective. You heard about SMA from Jeff. It is a muscular atrophy, a spinal muscular atrophy. There's a gene mutation that's been known for a while. And until 10 years ago, there weren't any disease-modifying therapies for SMA. And currently, there's not only a small molecule, there's also an antisense oligonucleotide and a gene therapy. And these currently live together pretty well in harmony to give options to patients. And I think this should be the goal for every group and every gene, to be able to have a diverse set of tools to treat the disorder, because not everything, as Jeff said, will work for everyone. And kind of following up on what Jeff was showing, I have here the 3 different drugs that are available for SMA. The small molecule that was actually the last to be approved, the antisense that was approved in 2016, and the AAV gene therapy that was approved in 2019. So everything happened in the last 6 to 7 years. And you can see that there are big differences between them, and the mechanism complements each other, because the AAV gene therapy restores the protein by giving the missing gene. The ASO makes an alternative gene make protein, and the small molecule is a splicing modifier that also works on the genetic basis of the disorder. So all 3 of them work well. Price-wise, as Jeff indicated, the small molecule is likely the cheapest, and the AAV gene therapy is likely the most durable therapy of the 3. So continuing on this line of comparing the small molecules to gene therapies and to ASOs, I highlighted a few things. I won't go through the whole table here, but I highlighted a few things that I thought were important. Small molecules, again, and Jeff, I'll constantly quote you. I hope it's okay. As Jeff said, they're the lowest hanging fruit if you're repurposing. They're a little bit more complicated if you want a brand new chemical entity that will do something that other compounds have not done before. But in general, they're very rarely disease modifying. It's very hard to find something that targets exactly where the mutation is and changes the origin of the disorder. If you're screening for and trying to repurpose, you don't always know how the small molecule is working, depending on how you're testing to look for the molecule. And it has on and off targets effects. So there are a lot of side effects that are associated with small molecules. The gene therapy advantage is it's a one-time therapy, but still we cannot redose. And people that have pre-existing antibodies to the virus cannot be dosed at all. So there's a barrier to treat certain patients, and definitely a big barrier to treat again in case you need a booster of the therapy. And antisense you heard a lot about, so I'm just going to skip. And if you have any questions to Jeff or me at the end, then feel free to ask us. So we know the target. What else do we need to know? So here I tried to give a little bit of a flavor of your child got a diagnosis, you got a diagnosis, you Google your gene, you look it up, you want to see what the protein's doing, and what do you need to know in order to go to someone and ask them to develop a therapy, or what does that someone need to know? You need to know, for gene therapy, it's really important to know what the size of the mRNA is, because that's what you're going to be putting into the virus, and that's what will make the protein when you're trying to replace it in your body. And not every mRNA fits in a cassette that goes into a virus. For example, in Duchenne muscular dystrophy, the gene is huge. So the trick there was to make a mini gene. And this is what raises the question whether if you cut and truncate the protein and it's not the original protein, is it really acting the same as the original protein? And this will be the question for many of the genes that were truncated and put in as a mini gene. You need to know what the introns and exons are for the gene. You need to know how the expression is regulated, especially if you want to look at upregulation and you want to know how your gene is modulated. You need to look at microRNAs or long non-coding RNAs. Jeff mentioned NMD and exon skipping. So all of these things you probably can just feed into a computer, and that will tell you a lot of the regulatory elements that are associated with the gene that you want to restore its function. And you want to know homology across species, and we learned the hard way when you have a gene that's very homologous between mouse and human and monkey, and you express it in the mouse and you express it in the monkey, and you want to see if you restored expression. If the gene is very, very similar among species, it's very hard to tease out the new expression versus what the animal is expressing in the background. So a lot of times we actually recommend to groups to optimize the human sequence so can be detectable over the animal background. What type, and you look at the mutated proteins, how is it mutated? How is it inherited? Is it a de novo mutation? Is it brand new in the family? No one knew before that anyone else suffers in the family from this mutation or carries the gene. That's important because if it is inherited, you want to go back and look at the mode of inheritance and find other patients that are potentially affected in the family. You want to know if it's a loss of function or gain of function. That's really important when you're designing a gene therapy or an antisense oligonucleotide. You want to know is there a genotype-phenotype correlation? If it's just a missense mutation, does it behave differently from a nonsense mutation when there's no protein at all? Or are the mutations all the same and they give the same clinical phenotype? This is really important when you start thinking about enrolling in a clinical trial. What are you looking for? What patients will you enroll in the trial based on the mechanism of therapy that you have decided to pursue for this indication? And then the last one I mentioned already, do you really need to know the protein function? As I said, I would argue that you don't have to know exactly what it does, but it's nice to know it if obviously there is information about it. What is proof of concept? And this is something that we're asked a lot by patient advocacy groups who want to partner with industry. What does industry want to see? So obviously it's not a one size fits all. Ideally, you want to see in vitro data, and it could be from 1 dimension, 2 dimension, 2.5 dimensions, or 3 dimensions. And if there is a good mouse model, we'd like to see data from a mouse model. Many groups have several different mouse models. It has to be the one, obviously, that's the most suitable for the modality that you've chosen. Cells could be either patients or isogenic cells, and organoids are now becoming more and more common, and especially for certain disorders and certain tissues, the data from the organoids is becoming really key for advancing things further into the clinic. And then the mouse model, a lot of times we have questions about, oh, we have a mouse model, but it doesn't behave like the humans. Is that okay? Well, obviously, ideally, we'd like to see some behaviors that are shared, but we want to know that the drug is working. So if your child has seizures, but the mouse does not seize, but the mouse has a different behavioral abnormality, if you can restore that, that's good enough. Or if you can show that the gene therapy or the ASO actually restore the mRNA and the protein, that's good enough. In the right place, that's good enough. So don't get stuck on the fact that the mouse doesn't behave like the humans. And there's always a question of when do you treat the mice? Everybody does P0 because it's really, P0 is immediately after the mice are born and the babies are tiny, tiny, and their skull is very, very soft. So it's easy to inject into the skull. And people tend to do that, but a P0 mouse does not represent a newborn or a patient post-birth, a human post-birth. So a lot of times people get start insisting on seeing data from a 3-week-old mouse or a 6-week-old mouse. Showing in P0 is a good start. If your drug does not work in P0, it's likely not going to work in a later age as well. And of course, we need biomarkers, and I encourage every group and every company that's working on disorders to start looking for biomarkers, because having an early indication that the drug is working is really helpful. So I wanted to give this example quickly of what we had when we in-licensed the program for a disorder called WORI. It's a very, very rare disorder. I think there are 200 patients known in the world, plus minus. The kids, a lot of them die. So the number probably is around 200 around steady state. It's inherited, it's an autosomal recessive, and the key feature of this disorder is very, very severe seizures, up to hundreds of seizures a day, and the kids end up passing away because of complications of the seizures. So when the PI came to us with work that was funded by the advocacy group, what he had at the time, and it's all been published since then, was a good mouse model that replicated some of the human features, an AV9 gene therapy under the SYN promoter. So the protein is expressed in neurons, which is where we need it to be expressed. And he was able to show that the gene therapy reverses all the symptoms that are seen in the mouse. So early mortality, failure to thrive, hypoglycemia, seizures, other things that were observed in the mouse were reversed by the gene therapy. And we thought to ourselves, you know, there are only 200 patients in the world. That's true. However, this gene therapy looks very compelling and we have to get it to patients. The proof of concept package was very robust and we decided to bring the program into Mozzie. Quickly, a few words about patient groups. You heard from some really great groups in the morning, and obviously great examples for what patient advocacy groups are doing these days and how advanced and how sophisticated the work is. On the left, you can see Cure Duchenne, the Cystic Fibrosis Foundation, and more recently FAST, the Angelman organization, that have been very successful by funding therapies. They're able to see a return on their investment and become very, very rich and very affluent and very influential advocacy groups. I just learned last week at a dinner that the Cystic Fibrosis Foundation has more than $3 billion under management, which is crazy, right? Everyone's dream to have so much money to be able to spend on your disorder. On the right-hand side is just a sample of the new CNS disorder groups that we're working with, and there are tens of them. And they're all hoping to be as successful as the ones on the left. So on sorry if I confused my right and left, but on the left is FAST, Cystic Fibrosis and Cure Duchenne, and on the right is are the new groups, and they're just aspiring to be as successful, but they don't have the resources, and this is where Mozzie comes in and hopes to support these groups and help them get into clinical studies and prove that the drugs that they're making are actually working and could be commercialized. So Armel showed a really great slide on the tools that their foundation has. And I built this pyramid of all the information that advocacy groups can actually generate to, first of all, have a strong basis for clinical development, and also to attract industry, because all these tools are already available and the industry partner does not have to spend money on registries or natural history studies or whatnot. So having all of this is really valuable. It costs money, true. So what would be the priorities? The priorities would be things that are maybe started with things that are less expensive, which is serving the community, understanding the disorder better, putting in place a disease concept model, which says what the priorities of the community are, versus spending a lot of money on looking for biomarkers, for example. But if you have the resources and you are able to do it, then everything on the bottom of the pyramid here could be executed by patient advocacy groups. As I said, registries, natural history studies, disease concept models, starting to think about clinical endpoints and the clinical endpoints that are meaningful in case your disorder has never been in clinical trials before, and in case you have some unique features and unique clinical symptoms that haven't been measured before, helping the industry partners and regulators understand what these endpoints are and how they're measures is really important. And I think that patient advocacy groups are best equipped to actually design those clinical endpoints. Looking for biomarkers, is there anything in the blood, in the CSF, in urine that could be any imaging or EEG biomarker that could be adopted for early response to therapy? That's very helpful. And then if you go up the pyramid, looking at what Armel showed in her slide earlier today, having cells, having organoids, having a biobank that also came up this morning, and having animal models, the more the ones that could be used for drug development, the better. And then on the right-hand side, I would say that's where industry comes in. We can guide proof of concept. What should it look like in vivo, in vitro? What are the IND enabling studies that are needed? What toxicology studies are needed? What would a clinical trial look like? How many patients will we enroll? What would be the endpoints? How many visits does the patient need to have in order to get the therapy and to measure the response to therapy? And then the choice of the modality will be based on the expertise of the company. So you'll go to Everyone or Lorem or La Jolla Labs if you want an ASO, or you go to other companies for a gene therapy and discuss with them what is the most suitable for your gene and your genetic mutations. Clinical development assumes that many of the disorders that we're working on have had limited previous clinical trial exposure before, and that we would like ideally to work on disease-modifying therapies. So, as I said before, small molecules not likely to be disease-modifying. ASOs, gene therapies, gene editing, of course, will likely be disease-modifying, and an intervention will change the fate of the patient and the trajectory of the disorder. The challenges are to validate novel endpoints or to modify known endpoints to be suitable for your disorder. The endpoint has to be sensitive enough to measure change. And sometimes when a population is very diverse, it's hard to find something that could measure across a population. The studies are small. In most cases, especially in gene therapy, we do not want a placebo or a sham control because it just doesn't make sense to bring a very fragile patient in and have them go through the whole procedure of injecting something into their brain or something into their liver, and then they're not getting anything, and then bringing them in again when they are eligible for treatment. So actually the regulatory agencies are more open today to running especially gene therapy or viral vector studies without a sham control or without a placebo. And then sometimes you have advisory boards and you hear from the patients one thing, from the clinicians a second thing, and from the regulatory agencies a third thing. How do you collate everything and make sure that you're going into the clinic with something that will satisfy the needs of the patients on one hand, but actually could be approved on the other hand? And again, biomarkers, that my head of regulatory brainwashed me with a biomarker part. Quickly on disease concept models, we start with caregiver surveys, interview families to understand what they would like to see changed. We go through a medical chart review to see what information is already available without needing to go into a prospective trial. We then interview the families again in a more qualitative manner. So go deep into interviews, understanding what a day in their life looks like. And based on all this information, we design a natural history study. And all this is put together to get a conceptual model that will guide us on what the clinical trial would look like and what the endpoints are. And all this is done in parallel. So nothing should be done isolated. All this should be parallel tracked, of course. So whatever you can do and jumpstart in the beginning, you're better off later on when you want to get into the clinic. And just going back to the Mazi slide, I wanted to show that the interaction between academics and family groups is critical. And I think it will become even more critical if less grants will be available from the NIH, or there will be restrictions on federal money. A lot of academics will be relying on patient advocacy group money to do work in their labs. And I hope this won't create more conflict of academics needing to publish and to get tenure while families really want to get drugs into the clinic and treat their loved ones. It's a joint effort. There should be alignment on goals and the same sense of urgency. And it's a process, and especially for young investigators that never worked with patient advocacy groups or families before, it is a shift in thinking. And families need to understand that usually when somebody went to academia to head a lab, developing a therapy was not the first thing that they had in mind when they signed up for the academic career. And the collaboration is based on one hand, the scientific expertise of the academics and everything that they bring to the table, with the acknowledgement of the fact that the advocacy groups bring a lot of expertise that otherwise they will not have access to, the academics will not have access to if they don't work hand in hand with the advocacy groups. So in summary, it takes a village to develop a therapy, and alignment, collaboration, synergy, however you want to call it, it's critical for this whole process. And we need solid biology, very smooth handovers between academics into biotech, with working hand in hand with the advocacy groups. So imagine a 4 by 100 race where somebody drops the baton and loses the race because of that. We don't want that to happen. We need to parallel track, so run everything as smoothly as we can at the same time and collaborate. So thank you, and happy to answer any questions you may have.
I have a microphone that helps too. And any questions? There's one.
I have two questions. So my first question is about the ASO modification, and both Jeff and Nadina, sorry, mentioned in your presentations. So there are various types of modification, including like LNA and MOA, or MOE, or GAPMER. So I want to hear your perspective or rationales how to choose the modification, pros and cons for each different type. So that's my first question, ASO modification. The second question is more for Nadina. So would you mind to elaborate a little bit more regarding how Lauren's team access the off-target effect for your ASO hits?
The regulatory experience, at least the feedback that we're getting from some of the ex-US regulators, is the commonality of backbone is something that they would actually really like to see if they're going to approach this more like a platform approval. It doesn't mean every drug just gets automatically approved. It just means they can truncate the amount of data that's needed for the review. And they've at least signaled early on that things like LNAs, which does create better stability, they actually mentioned that in their feedback to us, that they would rather not have different backbones, that would require a different level. So we're somewhat erring on the side of let's just get through what we can, even though I think we would really enjoy the opportunity to put an LNA in there. But that's just kind of one example where their feedback is that the commonality of backbone gives them more of a predilection to say, yeah, this is a truncated review based on the amount of repeatability you can show.
Yeah, I'm just going to add to that, that essentially there are a lot of years of experience now where we kind of have nailed, well, I'm going to use the word nailed, but I'm not sure if that's the correct word, the chemistry for GAPMERs and also for splice switching ASO. So for example, essentially for that, we know that for research purposes, so if you're in a lab and you want to use LNAs, that's completely fine. They're for clinic. We do know that these modalities are a little bit more toxic. So in the terms of the backbone, phosphothiorate is something that we routinely use, and especially in a GAPMER, I'm going to go technical here, in a GAPMER for CNS, a mixture of phosphothiorate with a phosphodiester is the one that we have the most knowledge on and the most results on. 2'-methoxyethyl modified on the RNA. This is also something that we have a lot on. And, also the splice switching, and Jeff can elaborate more on that, that is fully modifying an 18-mer, and essentially the backbone is mainly phosphothiorate. This is also something that has been now well established. So I think that they are, and the reason, as I was saying, that we use the chemistries is you want a drug, an oligo or whatever, to last long, to have less immunogenicity, right? To have high affinity for your RNA, which is essentially your target. And lastly, you want the best pharmacodynamic properties. So all of this together, this is basically what is on your tick list in order to define the best chemistry. And these chemistries are pretty much now defined for, at least for the ASOs.
Yeah, I mean, 250,000 people dosed with that chemistry. So that's a platform that has been tried and true. It's tested in many ways in the clinic. So that's basically it. That's where we focus. And at Ionis, we did a lot of work around, understanding the biology first and then teasing out the chemistry if we have to. So the idea was to find the target space, and then if you have to do a chemical SAR around a particular spot to tease out toxicity or improve potency, then that's kind of the strategy we took. I mean, for rare disease, it's just the clinical proof of concept. What's the fastest or the cheapest way to kind of get through the path? And if you change that chemistry, you throw a big wrench into the equation.
Okay. More questions here?
The question for Adina or Jeff. My understanding is that this, for ASOs, the delayed tox is something that's, it adds a lot of time to the development, but is also a little bit hard to predict from the sequence. Do you have any idea, what may be leading to that? And are we getting towards, some predictiveness for that?
It's a great question. It is a really good question. So I think and I'm going to share my perspective, there are twofold to this question. There are aspects of it that we can predict, and there are aspects of it of like some motifs in sequence that, you can build into your algorithms and predict that some of the things could potentially cause toxicity. And especially you would see that in the tolerability, which is, a smaller scale tox study before going to GLP tox. So that is something that, the more learnings we have, yes, we can fish some out and just, either cytotoxicity or immunogenicity. These are the two main things. But there are also some other aspects that at the moment we cannot predict them. So at the moment there is a molecule that can go to tolerability. And that's why I think, from my perspective, these toxicology studies are very, very important. Yes, they're costly and they're lengthy, but this is why they're important, because at this moment in time, there are some that we can predict, but there are some that we think that is not immunogenic, is super safe, not cytotoxic. I can't see anything wrong with it. Cells tolerate it just fine. And then we go to rats, and then they are either, acute FOB or delayed FOB. So there are things that we cannot predict. There are things that, and at least at Lorem, we really try to look at the data and see for any commonalities, because obviously having less of these leads into tolerability, we don't want to lose any good drug. But at the moment, this is a work in progress. So some we can predict, some not.
Yeah, and I'll just add to that it comes down to the data. I mean, there's some really good models that Ionis has developed, and but having delayed tox data is harder and longer and more expensive, and that's really what it comes down to. Once that data is there, then yeah.
Okay, we have two questions from the outside. The one is a more general one. Is there a centralized database that collects all the genetic data from rare disease patients around the world?
Unfortunately, no. We don't even have a centralized database in this country, but there are a number of countries across the world that are putting in, I would say, yeoman's work to try to create databases that are largely newborn screening. The Genomics England database is, I think, well over 100,000 at this point. Emirati and the Qatar genome projects, actually, I think Emirati alone is almost 1 million full. In Qatar, I think, is somewhere around 250,000. There's about 4 or 5 states, I know California is one of the states, where there is upwards of tens of thousands of newborn screening with whole genome sequencing, but there's not even a database in our country to do that. And I think, as I maybe alluded in my talk, wow, that is a fantastic effort for us to do for so many different reasons. And I think it opens up an entirely new ability for us to target disease. there's going to be issues with that too. I mean, one of the things that I noticed at Arab Health just a few weeks ago in Dubai is that they are incorporating newborn screening with genetic counseling and premarital counseling as well. And that's a concept that is going to be something to tackle as we get closer and closer towards having this evidence of gene and the number of mutations people may carry.
So if I can comment, there are, is that okay if I comment? I don't know if it's.
Yeah, absolutely. Go ahead.
There are, the issue is with data privacy, obviously, and different countries handle data privacy very differently. The Scandinavian countries have really good genetic databases, as the countries that Kent mentioned. But for example, if a company wants to use them, they have a very hard time accessing this information. If only mutation information is needed, then ClinVar is probably the best source for genetic information that's totally de-identified and just has information about mutations in genes.
Okay, thank you. I have one more question online and then maybe one more in the room, and then we have to move on. Oh, there are two maybe. So one that came in online is, are there any challenges for ASO administration that goes intrathecal?
Yeah, I mean, there's always going to be challenges anytime you're in that space. I'll just say from a research perspective, it's challenging from the perspective that on the one hand, it's hard to find people that can actually do it well and do it well and consistently. So if you think about just actually doing that in a research setting and the consistency is a challenge, think about it in an actual treatment setting as well. And that's always going to be the concern with any kind of intrathecal administration. So the idea of accredited treatment centers, places that actually are defined in being able to do this, there's multiple disciplinary aspects of those treatment centers. What we see in research is what we're going to see in the real world settings as well. But it's not an ideal space. That's where, for me, I mean, we're intrathecal administration, of course, but I'm really hoping that there are companies that are going to come along with different modalities of delivery. I think it's going to take a while, though, unfortunately.
Yeah, and just to add to that, that's why I was saying that having really, really good physicians and really good treatment centers, as you were saying, that they're really equipped. I mean, of course, you require anesthesia a lot of times because of the, the specific challenges of these patients and dosing them intrathecally. However, having said that, it is a route of administration that is well established, and unfortunately at the moment, until we have another, exactly, and a safe one, and until we have another delivery, this is.
We have one more question.
Yes. Thank you. Well, first of all, thank you all for the session. It was fantastic. So my question is on behalf of my husband, Bernie Ziprich. So Kent, you talked about the need for a better regulatory pathway, but N-Lorem seems to be finding success with the current FDA framework. Can you help us understand the nuances better and some of the changes you're hoping to achieve?
Yeah, it's a great question, because I think in some respects, folks may see as N-Lorem and Everyone Medicines and others as sort of in competition. We don't see it as competition. I mean, I think the one stark difference between what N-Lorem's doing versus Everyone Medicines is that we're actually just going down the route within Europe to create a brand new pathway. And I think N-Lorem is actually going with the existing pathway, but using that totality of data to also negotiate a new pathway while going down the existing one. Our attempt is to really try to create as much data as we can in this sort of compendium approach. And the thinking behind it is that the timing right now, at least in Europe, seems to be very favorable. They clearly understand you can't do a clinical study. The question of GLP tox, quite honestly, even if they require that, if it's a GLP tox that's reasonable and that we can figure out a way to make it even somewhat affordable, that's okay. But right now, there's not a pathway that says it's not only approved for marketing authorization, but then it'll get reimbursed as a result. I think we're all kind of trying to bring as much as we can to bear to give them the experience to say, yeah, this is how to do it. And this is the way to go about it from a research perspective. And it's not just for research, it's for treatment and approval. Reimbursement's the key here.
All right, thank you.